Hormones are important substances for life. Insulin is one of those hormones. Now, preparations are being made for a series of experiments to investigate the mechanism of insulin secretion. This is the pancreas. The pancreas looks white because of its exocrine tissue. The blood vessels run through the central part of the tissue. Besides these vessels, there are some translucent round objects. These are the endocrine tissues, the islets of Langerhans. Aldehyde thionin, a violet pigment, and red and green pigments were used to stain the pancreas. Then the exocrine tissue was stained red. The islets of Langerhans, stained violet, are clearly observed in the exocrine tissue. These violet cells are called B cells, and they secrete insulin. About 80% of the total tissue is dominated by these B cells. Surrounding the B cells, there are one or two layers of A cells. Now let us look at these cells with the electron microscope. The A cells are on the right-hand side. These granules contain glucagon. On the left, there are B cells, which contain insulin. The core of the granule of the B cell often shows such a crystalline structure. The repeated regular stripes seen in the crystal may indicate the orderly arrangement of insulin molecules. Now a primary culture of the pancreatic cells is being made in order to investigate the mechanism of insulin secretion. Our final aim is to observe the B cells of the islets of Langerhans in this culture. The pancreas obtained from 4-day-old rats is cut into very small pieces. These pieces are washed in physiological saline. Then a solution of trypsin and collagenase is added. The temperature is kept at 37 degrees centigrade. The pancreatic tissue is gradually dissociated. The isolated cells are suspended in a culture medium and poured into sterile Petri dishes. The carbon dioxide incubator. In this apparatus, the B cells can survive without losing their original functions, even after they've been separated from pancreatic tissue. 72 hours have passed. The cultured cells have spread out. In this condition, their internal structures can be easily observed. The cells of the islets of Langerhans look dark, as they contain secretory granules. Besides B cells, the colony contains other kinds of endocrine cells. Let us apply aldehyde thionin stain to this cell colony. The B cells are stained blue. Magnifying the B cells further, the beta granules appear to move actively. These beta granules were stained with aldehyde thionin. Now it becomes clear why the cells looked blue when stained with that pigment. This time, let us pursue the behavior of the beta granules by adding glucose and/or tolbutamide, which stimulate insulin secretion. Glucose is physiologically an important stimulant for insulin secretion. Tolbutamide, on the other hand, is a sulfonylurea effectively used for diabetes treatment. The behavior of the beta granules is recorded with time-lapse photography. The beta granules, which have been evenly spread out in the cytoplasm, start to show some changes in their distribution. Two hours after addition of tolbutamide, the beta granules come up to the cell membrane and line up along it. This time, let us add glibenclamide, which is a recently developed sulfonylurea. Now some changes are observed in the B cells on the left-hand side. The beta granules have begun to come up to the cell membrane. The same changes have occurred as were observed when tolbutamide was added. Now let us see how the beta granules are formed. There is a Golgi complex beside the nuclei on the upper right-hand side of the picture. A part of the entangled stick-like structure has become swollen. Soon that part is torn off from the structure. This is the beta granule. The beta granules thus formed come out of the Golgi complex and pass into the cytoplasm. The beta granules move actively on their way through the mitochondria. The granules, about 0.2 micron in diameter, contain within them an important material for our normal lives. We are now observing their behavior. This is the electron microscopy of the granules. The beta granules exist in the cytoplasm. The granules which are adjacent to the cell membrane. These are the granules just beginning to secrete. The moment of insulin secretion. Now let us add caffeine to the B cells. Caffeine stimulates insulin secretion by increasing the cyclic AMP in the cell. The cell becomes swollen. The beta granules gather on the inside of the cell membrane and line up with it, as we have seen when a sulfonylurea was used. The islets of Langerhans. This small organ plays an important role in maintaining the homeostasis of our bodies. Here are the B cells which exist in these small islets. We have been observing smaller entities called beta granules. Researchers are continuing their study today in order to investigate the mechanism of these tiny objects in our bodies.